Dear learners, in this video we are going to study chapter 9 biomolecules of class 11th NCERT biology. So the read with us the chapter because reading is the first step of good learning. Here we start. There is a wide diversity in living organisms in our biosphere. Now a question that arises in our minds is, are all living beings made of the same chemicals? It means elements and compounds. You have learned in chemistry how elemental analysis is performed. If we perform such an analysis on a plant tissue, animal tissue, or microbial paste, we obtain a list of elements like carbon, hydrogen, oxygen, and several others, and their respective content per unit mass of a living tissue. If the same analysis is performed on a piece of Earth's crust, as an example of non living matter, we obtain a similar list. What are the differences between the two lists? In absolute terms, no such differences could be made out. All the elements present in a sample of Earth's crust are also present in a sample of living tissue. However, a closer examination reveals that the relative abundance of carbon and hydrogen with respect to other elements is higher in any living organism than in Earth's crust. Moving to our next heading, that is how to analyze chemical composition. We can continue asking the same way, what type of organic compounds are found in living organisms? How does one go about finding the answer? To get an answer, one has to perform a chemical analysis. We can take any living tissue, a vegetable or a piece of liver, etc., and grind it in trichloroacetic acid Cl3CCOOH using a mortar and a pestle. We obtain a thick slurry. If we were to strain this through a cheesecloth or cotton, we would obtain two fractions. One is called the filtrate, or more technically the acid soluble pool, and the second retintate or acid insoluble fraction. Scientists have found thousands of organic compounds in the acid soluble pool. In higher classes, you will learn about how to analyze a living tissue sample and identify a particular organic compound. It will suffice to say here that one extract the compounds, the subject extract to various separation techniques. Still, one has separated a compound from all other compounds. In other words, one isolates and purifies a compound. Analytical techniques, when applied to the compound, give us an idea of molecular formula and the probable structure of compound. All the carbon compounds that we get from living tissues can be called biomolecules. However, living organisms have also got inorganic elements and compounds in them. How do we know this? A slightly different but destructive experiment has to be done. One weighs a small amount of a living tissue, say a leaf or liver, and this is called wet weight, and dry it. All the water evaporates. The remaining material gives dry weight. Now, if the tissue is fully burned, all the carbon compounds or oxidized to gaseous form, CO2, water vapor, and are removed. What is remaining is called ash. This ash contains inorganic elements like calcium, magnesium, etc. Inorganic compounds like sulfate, phosphate, etc. are also seen in acid soluble fraction. Therefore, elemental analysis gives elemental composition of living tissues in the form of hydrogen, oxygen, chlorine, carbon, etc. While analysis for compounds gives an idea of the kind of organic and inorganic constituents present in living tissues. From a chemistry point of view, one can identify functional groups like aldehydes, ketones, aromatic compounds, etc. But from a biological point of view, we shall classify them into amino acids, nucleotide bases, fatty acids, etc. Amino acids are organic compounds containing an amino group and hence acidic group are as substituents on the same carbon. It means alpha carbon. Hence they are alpha amino acids. They are substituted methanes. There are four substituent groups occupying the four valency positions. These are hydrogen, carboxyl group, amino group, and variable group, designated as R group. Based on the nature of R group, there are many amino acids. However, those which occur in proteins are only of 20 types. The R group in these proteinaceous amino acids could be hydrogen. The amino acid is called glycine. A methyl group, alanine, hydroxyl methyl serine, etc. 3 of the 20 are shown in figure 9.1. The chemical and physical properties of amino acids are essentially of the amino, carboxyl, and are functional groups. Based on number of amino and carboxyl group, there are acidic, example glutamic acid, basic lysine, and neutral valine amino acids. Similarly, there are aromatic amino acid, tyrosine, phenylalanine, tryptophan. A particular property of amino acids is the ionizable nature of minus NH2 and minus COOH groups. Hence, in solution of different pH, the structure of amino acids changes. Like here, B is called zwitterionic form. This is the CH. This is CH. And one bond is NH3, one bond is from COOH, and one is the functional group. This is A, this is B, and this is C. Here, CH make bonds with COO and with NH3, then a functional group R. CH make here the CH makes bond with NH2 with here with the COO, and on the upper side with R, a functional group. Lipids are generally water insoluble. They could be simple fatty acids. A fatty acid is a carboxyl group attached to an R group. The R group could be a methyl minus CH3 or ethyl minus CH2H5 or a higher number of minus CH2 groups, 1 carbon to 19 carbons. For example, palmitic acid has 16 carbons including carboxyl carbon. Arachidonic acid has 20 carbons atoms including the carboxyl carbon. Fatty acids could be saturated without double bond or unsaturated with one or more C double bonds. Another simple lipid is glycerol, which is trihydroxypropane. Many lipids have both glycerol and fatty acid. Here the fatty acids are found esterified with glycerol. They can be then monoglycerides, diglycerides, and triglycerides. These are also called fats and oils based on the melting point. Oils have lower melting point, example gingerly oil, and hence remain as oil inventors. Can you identify fat from the market? Some lipids have phosphorus and phosphorylated organic compound in them. These are phospholipids. They are found in cell membrane. Lecithin is one example. Some tissues, especially the neural tissues, have lipids with more complex structures. Living organisms have a number of carbon compounds in which heterocyclic rings can be found. Some of these are nitrogen bases: adenine, guanine, cytosine, uracil, and thymine, that are called as ATCG. Here we will continue. When found attached to a sugar, they are called nucleosides. If a phosphate group is also found esterified to the sugar, they are called nucleotides. Adenosine, guanosine, thymidine, uridine, cytidine are nucleosides. Adenylate acid, thymidylate acid, guanylic acid, uridylic acid, and cytidylate acid are nucleotides. Nucleic acids like DNA and RNA consist of nucleotides only. DNA and RNA function as genetic material. Here, the diagram of glucose, that is in hexagonal. Here is the diagram of ribose. This is glycine, this is alanine, and this is serine, and these are amino acids. And these are sugars and or carbohydrates. Here, this is palmitic acid, this is glycerol, this is triglyceride. R1, R2, and R3 are fatty acids here. This is phospholipid, lecithin. This is cholesterol. This is adenine, purine, adenosine. This is adenylic acid. This nucleoside, that is uridine, nucleotide, and uracil, pyrimidine, nitrogen bases. Here we go further and move on our next topic, that is primary and secondary metabolites. Primary and secondary metabolites. The most exciting aspect of chemistry deals with isolating thousands of compounds, and small and big, from living organisms, determining their structure and if possible synthesizing them. If one were to make a list of biomolecules, such a list would have thousands of inorganic compounds including amino acids, sugars, etc. For reasons that are given in section 9.10, we can call these biomolecules as metabolites. In animal tissues, one notices the presence of all such categories of compounds shown in figure 9.1. These are primary metabolites. However, when one analysis plant, fungal, and microbial cells, one would see thousands of compounds other than these primary metabolites. Example: alkaloids, flavonoids, rubber essential oils, antibiotics, colored pigments, scents, gums, spices. These are called secondary metabolites. While primary metabolites have identifiable functions and play known roles in normal physiological processes, we do not at the moment understand the role or function of all the secondary metabolites. In host organisms, however, many of them are useful to human welfare: rubber, drugs, spices, scents, and pigments. Some secondary metabolites have ecological importance. In the latter chapters and years, you will learn more about this. Next heading is biomacromolecules. There are one feature common to all those compounds found in the acidic soluble pool. They have molecular weights ranging from 18 to around 800 Daltons approximately. The acidic insoluble fraction has only four types of organic compounds. It means proteins, nucleic acid, polysaccharides, and lipids. These classes of compounds, with the exception of lipids, have molecular weights in the range of 10,000 Daltons and above. For this very reason, biomolecules, it means chemical compounds found in living organisms, are of two types. One, those which have molecular weights less than 1000 Dalton and are usually referred to as micromolecules or simply biomolecules, while those which are found in the acid insoluble fraction are called macromolecules or biomacromolecules. The molecules in the insoluble fraction, with the exception of lipids, are polymeric substances. Then why do lipids, those molecular weights do not exceed 800 Dalton, come under acid insoluble fraction? It means macromolecular fraction. Lipids are indeed small molecular weight compounds and are present not only as such but also arranged into structures like cell membrane and other membranes. When we grind a tissue, we are disrupting the cell structure. Cell membrane and other membranes are broken into pieces and form vesicles which are not water soluble. Therefore, these membrane fragments in the form of vesicles get separated along with the acidic insoluble pool, and hence the macromolecular fraction. Lipids are not strictly macromolecules. The acid soluble pool represent roughly the cytoplasmic composition. The macromolecules from cytoplasm and organelles become the acid insoluble fractions. Together, they present the entire chemical composition of living tissues or organisms. In summary, if we represent the chemical composition of living tissue from abundance point of view and arrange them class wise, we observe that water is the most abundant chemical in living organisms. Next heading is proteins. Proteins are polypeptides. Here I will change the color. They are linear chains of amino acids linked by peptide bonds as shown in figure 9.3. Each protein is a polymer of amino acids, as there are 20 types of amino acids. Example: alanine, cysteine, proline, tryptophan, lysine, etc. Protein is a heteropolymer and not a homopolymer. A homopolymer has only one type of monomer repeating n number of times. This information of the amino acid content is important as later in your nutrition lessons you will learn that certain amino acids are essential for our health and that they have to supply it through our diet. Hence, dietary proteins are the source of essential amino acids. Therefore, amino acids can be essential or non-essential. The latter are those which our body can make. While we get essential amino acids through our diet or food, proteins carry out many functions in living organisms. Some transport nutrients across cell membranes, some fight infectious organisms, some are hormones, some are enzymes, etc. Collagen is the most abundant protein in animal world, and ribulose have biphosphate carboxylase oxygenase are ribulose bisphosphate carboxylase oxygenase is the most abundant protein in the whole of the biosphere. Next is polysaccharides. The acid-insoluble pellet also has polysaccharides, carbohydrates as another class of macromolecules. Polysaccharides are long chains of sugars. They are threads, literally a cotton thread, containing different monosaccharides as building blocks. For example, cellulose is a polymeric polysaccharide consisting of only one type of monosaccharide. It means glucose. Cellulose is a homopolymer. Starch is a variant of this but present as a storehouse of energy in plant tissues. Animals have another variant called glycogen. Inulin is a polymer of fructose in a polysaccharide chain, say glycogen. The right end is called the reducing end and the left end is called the non-reducing end. It has branches as shown in the form of cartoon. Starch form helical secondary structures. In fact, starch can hold I2 molecules in the helical portion. The starch minus I2 is blue in color. Cellulose does not contain complex helices and hence cannot hold I2. Here is a diagrammatic representation of portion of glycogen. Plant cellulose are made of cellulose. Paper made from plant pulp and cotton fiber is cellulosic. There are more complex polysaccharides in nature. They have building blocks, amino sugars, and chemically modified sugars. Example: glucosamine, glucosamine, and acetyl sorry, galactosamine, etc. Exoskeletons of arthropods, for example, have a complex polysaccharide called chitin. These complex polysaccharides are mostly homopolymers. Nucleic acids. The other type of macromolecule that one would find in the acid-insoluble fraction of any living tissue is the nucleic acid. These are polynucleotides together with polysaccharides and polypeptides. These comprise a true macromolecular fraction of any living tissue or cell. For nucleic acids, the building block is a nucleotide. A nucleotide has three chemical distinct components. One is heterocyclic compound. The second is a monosaccharide and the third is phosphoric acid or phosphate group. As you notice in figure 9.1, the heterocyclic compounds in nucleic acids are the nitrogenous bases named adenine, guanine, uracil, and cytosine and thymine. Adenine and guanine are substituted purine, while the rest are substituted pyrimidines. The skeletal heterocyclic ring is called purine and pyrimidine respectively. The sugar found in polynucleotide is either ribose, a monosaccharide, pentose, or 2-deoxyribose. A nucleic acid containing deoxyribose is called deoxyribonucleic acid DNA, while that would contain ribose is called ribonucleic acid called RNA. Next is structure of proteins. Proteins, as mentioned earlier, had heteropolymers containing strings of amino acid. Structures of molecules mean different things in different contexts. In inorganic chemistry, the structure invariably refers to the molecular formulae. Example: NaCl, MgCl2, etc. Organic chemists also always write a two-dimensional view of molecules while representing the structure of the molecules. Example: benzene, naphthalene, etc. Physicists conjure up the three dimensional views of molecular structures, while biologists describe the protein structure at four levels. The sequence amino acids, it means the positional information in a protein, which is the first amino acid, which is second, and so on, is called primary structure of a protein. A protein is imagined as a line at the left end represented by the first amino acid and the right end represented by the last amino acid. The first amino acid is also called N-terminal amino acid. Then last amino acid is called the C-terminal amino acid. A protein thread does not exist throughout as an extended rigid rod. The thread is folded in the form of helix, similar to a revolving staircase. Of course, only some proportions of the protein thread are arranged in the form of helix. In proteins, only right-handed helices are observed. Other regions of the protein thread are folded into the other forms in what is called secondary structure. In addition, the long protein chain is also folded upon itself like a hollow woolen ball, giving rise to the tertiary structure. This gives a three-dimensional view of protein. Tertiary structure is absolutely necessary for the many biological activities of proteins. Some proteins are an assembly of more than one polypeptide or subunit. The manner in which these individual folded polypeptides or subunits are arranged with respect to each other. Example, linear strings of spheres. Spheres are arranged one upon other in the form of cube or plate, etc. Is the architecture of a protein, otherwise called the quaternary structure of a protein. Adult human hemoglobin consists of 4 subunits. 2 of these are identical to each other, hence 2 subunits of alpha type and 2 subunits of beta type together constitute the human hemoglobin Hb. Enzymes. Almost all enzymes is a protein. There are some nucleic acids that behave like enzymes. These are called ribozymes. One can depict an enzyme by a line diagram. An enzyme can, like any protein, has a primary structure. It means amino acid sequence of the protein, and enzyme, like any protein, has the secondary and tertiary structure. When you look at a tertiary structure, upon itself, the chain crosses itself, and hence many crevices or pockets are made. One such pocket is the active site. An active site of an enzyme is a crevice or pocket into which the substrate fits. Thus, enzymes through their active site catalyze reactions at a high rate. Enzymes catalysts differ from inorganic catalysts in many ways, but one major difference need mention. Inorganic catalysts work efficiently at high temperatures and high pressures, while enzymes get damaged at high temperatures. However, enzymes isolated from organisms who are normally live under extremely high temperatures. Example, hot events and sulfur springs are stable and retain their catalytic power even at high temperatures. The thermal stability is thus an important quality of such enzymes isolated from thermophilic organisms. Next is chemical reactions. How do we understand these enzymes? Let us first understand a chemical reaction. Chemical compounds undergo two types of changes. A physical change simply refers to a change in shape without breaking of bonds. This is a physical process. Another physical process is a change in state of matter. When ice melts into water, or when water becomes a vapor, these are also physical processes. However, when bonds are broken and new bonds are formed during transformation, this will be called a chemical reaction. For example, BaOH2 plus H2SO4 gives BaSO4 plus 2H2O is an for example, this is an organic chemical reaction. Similarly, hydrolysis of starch into glucose is an organic chemical reaction. Rate of physical or chemical process refers to the amount of product formed per unit time. It can be expressed as rate equal P by T. Rate can also be called velocity if the direction is specified. Rates of physical and chemical process are influenced by the temperature among other factors. A general rule of thumb is that rate double or decreases by half for every 10 degrees Celsius change in either direction. Catalyzed reactions proceed rates vastly higher than that of uncatalyzed ones. When enzymes catalyzed reactions are observed, the rate would be vastly higher than the same but uncatalyzed reaction. For example, carbon dioxide, it means CO2 plus H2O water. When carbonic anhydrase gives H2CO3, that is called as carbonic acid. In the absence of an enzyme, this reaction is very slow, with about 200 molecules of H2CO3 being formed in an hour. However, by using the enzyme present within the cytoplasm called carbonic anhydrase, the reaction is speed dramatically with about 6 lakhs molecules being formed every second. The enzyme has accelerated the reaction rate by about 10 million times. The power of enzymes is incredible indeed. There are thousands of types of enzymes, each catalyzing a unique chemical or metabolic reaction. A multi-step chemical reaction, when each of the steps is catalyzed by the same enzyme complex or different enzymes, is called a metabolic pathway. For example, glucose to 2-pyruvic acid in Krebs cycle is C6H12O6 plus O2 gives C2C3H4O3 plus 2H2O. And it is actually metabolic pathway in which glucose become pyruvic acid through 10 different enzyme catalyzed metabolic reaction. When we study respiration in chapter 12, we will study these reactions. At this stage, you should know that this very metabolic pathway with one or two additional reactions give rise to a variety of metabolic end products. In our skeletal muscle, under anaerobic conditions, lactic acid is formed. Under normal aerobic conditions, pyruvic acid is formed. In yeast, during fermentation, the same pathway leads to the production of ethanol, alcohol. Hence, in different conditions, different products are possible. Next is how do enzymes bring about such high rates of chemical conversions. To understand this, we should study enzymes a little more. We have already understood that idea of active site. The chemical or metabolic conversion refers to a reaction. The chemical which is converted into the product is called a substrate. Hence, enzymes, that means proteins which with three-dimensional structure including an active site, convert a substrate into a product symbolically. This can be depicted as S to P. It is now understood that the substrate has to bind the enzyme at its active site within a given cleft or pocket. The substrate has to diffuse towards the active site. There is thus the obligatory formation of ES complex. E stands for enzyme. This complex formation is a transient phenomena during the state where substrate is bound to enzyme active site. A new structure of the substrate called transition state structure is formed. Very soon after the expected bond breaking or making is completed, the product is released from the active site. In other words, the structure of substrate gets transformed into the structure of products. The pathway of this transformation must go through the so-called transition state structure. There could be many more altered structured state between the stable substrate and the product. Implicit in this statement is the fact that all other intermediate structures state are unstable. Stability is something related to energy status of the molecule or the structure. Hence, when we look at this pictorially through graph, it looks like something as in figure 9.4. Here it is concept of activation energy. The y-axis represents the potential energy contained. The x-axis represents the progression of the structural transformation or states through the transition state. You will notice two things: the energy level different between S and P. If P is at lower level than S, the reaction is an exothermic reaction. One need not supply energy by heating in order to form the product. However, whether it is an exothermic or a spontaneous reaction or an endothermic or energy requiring reaction, the S has to go through a much higher energy state is transition state. The difference in average energy content of S from that of this transition state is called activation energy. Enzymes eventually bring down this energy barrier, making the transition of S to P more easily. Next is nature of enzyme action. Each enzyme has substrate as binding site in its molecule, so that a highly reactive enzyme substrate complex ES is produced. This complex is short-lived and dissociates into its product P, and the unchanged enzyme with an intermediate formation of enzyme product complex EP. The formation of ES complex is essential for catalysis. E plus S, ES gives EP gives E plus P. The catalytic cycle of an enzyme action can be described in the following steps. First, the substrate binds to the active site of enzyme, fitting into the active site. Second, the binding of the substrate includes the enzyme to alter its shape, fitting more tightly around the substrate. Third, the active site of enzyme now in close proximity of the substrate breaks the chemical bonds of the substrate and the new enzyme product complex is formed. Fourth, the enzyme releases the products of the reaction and the free enzyme is ready to bind to another molecule of substrate and run through the catalytic cycle once again. Next is factors affecting enzyme activity. The activity of an enzyme can be affected by a change in conditions which can alter the tertiary structure of protein. These include temperature, pH, change in substrate concentration, or binding of a specific chemicals that regulate its activity. Temperature and pH. Enzymes generally function in a narrow range of temperature and pH. Each enzyme shows its highest activity at a particular temperature, and pH is called optimum temperature and optimum pH. Activity declines both below and above the optimum value. Low temperature preserves the enzyme in a temporarily inactive state, whereas high temperature destroys enzymatic activity because of proteins are denatured by heat. Concentration of substrate. With the increase in substrate concentration, the velocity of enzymatic reaction rises. At first, the reaction ultimately reaches a maximum velocity maximum, which is not exceeded by any further rise in concentration of substrate. This is because the enzyme molecules are fewer than the substrate molecules, and after saturation of these molecules, there are no free enzymes molecules to bind with additional substrate molecules. The activity of an enzyme is also sensitive to the presence of a specific chemical that bind to the enzyme. When the binding of the chemical shuts off enzyme activity, the process is called inhibition and the chemical is called inhibitor. When the inhibitor closely resembles the substrate in its molecular structure and inhibits the activity of the enzyme, it is known as competitive inhibitor. Due to its close structural similarity with its substrate, the inhibitor competes thus with the substrate for the substrate binding site of enzyme. Consequently, the substrate cannot bind at. As a result, the enzyme action declines. Example: inhibition of succinic dihydrogenase by malonate, which closely resembles the substrate succinate in structure. Such competitive inhibitors are often used in the control of bacterial pathogens. Next is classification or nomenclature of enzymes. Thousands of enzymes have been discovered, isolated, and studied. Most of these enzymes have been classified into different groups based on the types of reactions they catalyze. Enzymes are divided into 6 classes, each with 4 to 3 subclasses and named accordingly by a 4-digit number. Oxidoreductases, dehydrogenases, enzymes which catalyze oxidoreductases or dehydrogenases, enzyme which catalyze oxidoreduction between two substrates S and S dash. Example: S reduced plus S dash oxidized, S oxidized plus X dash reduced. Transferases, enzyme catalyzing a transfer of a group Z other than hydrogen between a pair of substrate S and S dash. Example: S minus G plus S dash gives S plus S dash minus G. Hydrolysis, enzymes catalyzing hydrolysis of ester, ether, peptide, glycosidic, C bond C bond halide or P N bonds. Lyases, enzymes that catalyze removal of groups from substrate by mechanisms other than hydrolysis, leaving double bonds. Isomerases, including all enzymes catalyzing interconversion of optical, geometric or positional isomers. Ligases, enzymes catalyzing the linking together of two compounds. Example: enzymes which catalyze joining of C bond O, C bond S, C bond N, P bond O, etc. bonds. Cofactors, enzymes are composed of one or several polypeptide chains. However, there are a number of cases in which non-protein constituents are called cofactors are bound to the enzyme to make the enzyme catalytically active. In these instances, the protein portion of the enzymes is called the apoenzyme. Three kinds of cofactors may be identified: prosthetic groups, coenzymes, and metal ions. Prosthetic groups are organic compounds and are distinguished from other cofactors in that they are tightly bound to the apoenzyme. For example, in peroxidase and catalase, which catalyze the breakdown of hydrogen peroxide to water and oxygen. Heme is the prosthetic group and it's part of active site of the enzyme. Coenzymes are also organic compounds, but their association with the apoenzyme is only transient, usually occurring during the course of catalysis. The furthermore, coenzymes serve as cofactors in a number of different enzyme catalyzed reactions. The essential chemical components of many coenzymes are vitamins. Example: coenzyme in nicotinamide adenine dinucleotide NAD and NADP containing the vitamin niacin. A number of enzymes require metal ions for their activity, which form coordination bonds with side chains at the active site and at the same time form one or more coordination bonds with the substrate. Example: zinc is a cofactor for the proteolytic enzyme carboxypeptidase. Catalytic activity is lost when the cofactor is removed from the enzyme, which testifies that they play a crucial role in catalytic activity of enzyme. Summary: although there is a bewildering diversity of living organisms, their chemical composition and metabolic reaction appear to be remarkably similar. The elemental composition of living tissues and non-living matter appear also to be similar when analyzed qualitatively. However, a closer examination reveals that the relative abundance of carbon, hydrogen, and oxygen is higher in living systems when or compared to an inanimate matter. The most abundant chemical in living organism is water. There are thousands of small molecular weight, less than 1000 Dalton biomolecules, amino acid, monosaccharide, and disaccharide sugars. Fatty acids, glycerol, nucleotides, and nucleosides and nitrogen bases are some of organic compounds seen in living organisms. There are 20 types of amino acids and 5 types of nucleotide. Fat and oils are glycerides, in which fatty acids are esterified to glycerol. Phospholipids contain, in addition, a phosphorylated nitrogenous compound. Only 3 types of macromolecules, it means proteins, nucleic acids, and polysaccharides, are found in living systems. Lipids, because of their association with membranes, separate in micromolecule fraction. Biomolecules are polymers. They are made of building blocks which are different. Proteins are heteropolymers made of amino acids. Nucleic acid, RNA, and DNA are composed of nucleotide. Biomacromolecules have a hierarchy of structures: primary, secondary, tertiary, and quaternary. Nucleic acids serve as genetic material. Polysaccharide are composed components of cell wall in plants, fungi, and also of exoskeleton of arthropods. They are also storage forms of energy. Example: starch and glycogen. Proteins have a variety of cellular functions. Many of them are enzymes, some are antibodies, some are receptors, some are hormones, and some are other and structural proteins. Collagen is the most abundant protein in animal world, and ribulose bisphosphonate carboxylase oxygenase is the most abundant protein in the whole of the biosphere. Enzymes are proteins which catalyze biochemical reaction in the cells. Ribozymes are nucleic acids which catalytic power. Proteinaceous enzymes exhibit substrate specificity, require optimum temperature and pH for maximal activity. They are denatured at high temperatures. Enzymes lower activation energy of reactions and enhance greatly the rate of reactions. Nucleic acids carry hereditary information and are passed on from parental generation to the progeny. Here the chapter is finished, and so bye bye.